You can tweet.
Please do. Good morning, ladies and gentlemen. Thanks, Matt, for that very kind introduction. My interest is in metabolic syndrome and in polycystic ovary syndrome and type 2 diabetes. So I'm hoping with this kind of a very broad introduction and presentation, initially to engage as many of you as possible. And then afterwards, Manju is going to come up and talk about the application of his interest, which is looking at novel proteins, novel muscle proteins, to see whether or not you as a group potentially would like to collaborate with us and actually look at sports medicine and these markers, both in health and disease. All right then, so let's talk about polycystic ovary syndrome. Matt, how do I do this? That's it, that's great, that's fantastic. Okay, so polycystic ovary syndrome is a very common condition among the female population. Approximately 10% of the population of premenopausal females, and arguably the most important endocrine condition that we have. Patients with polycystic ovary syndrome have multiple symptomatology. They present to endocrinologists and diabetologists and gynecologists and obstetricians, dermatologists, and also to psychiatry because of eating disorders. And the annual economic burden in the US is around about $4.6 billion. And there's a wide range of presentations, and the actual diagnosis is under debate. So what I want to do first of all is say what the prevalence and what the problem here in Qatar. So we took 750 women between the ages of 18 and 40 from the Qatar Biobank. And of that, 720 had a testosterone SHBG. And basically, based on a raised androgen and irregular periods, one can actually diagnose PCOS either from the NIH criteria or from the Androgen Excess Society. And what we actually found is that 97 of 720 fulfilled that diagnosis of polycystic ovary syndrome. So that's around 13.5%. Now, if one takes the Rotterdam criteria, we look at polycystic ovaries on ultrasound, that's probably around about 20 to 21%. So that's double what we'd normally find in a Caucasian population. And you can say, well, so what? So let's talk about metabolic syndrome. As this audience probably knows, that metabolic syndrome is a kind of collection of metabolic risk factors that accelerate the onset of diabetes, heart disease, stroke, and certain cancers. And it was first termed Syndrome X by Reven in 1988, but it's also known as diabetes, insulin resistance syndrome, and cardiovascular dysmetabolic syndrome. It is a constellation, and there's three definitions, but I want to focus just on the IDF diagnosis, where primarily essential obesity. So first of all, one measures the waist of either males or females, and it's if you've got a waist measurement over 94 centimeters for men and 80 centimeters for women, plus any two of either 1, raised triglyceride levels, 2, a low HDL level, 3, raised blood pressure level of greater than 130 over 85, and 4, a raised fasting plasma glucose of 5.6 or greater. The prevalence of metabolic syndrome and PCOS is high. 43% of 106 patients had metabolic syndrome in the US, and in Europe it's around about 16%. So one looks at the number of risk factors that these patients with polycystic ovary syndrome have. At 90% of PCOS patients have at least one of those risk factors. And you can actually say, well, again, so what? Well, so what is shown here. And here's the cardiometabolic risk. So we know that cardiovascular disease mortality increases with a number of metabolic syndrome risk factors. And what you can actually see here, as even with 1 to 2 risk factors, the hazard ratio increases to 1.73, so a 73% increase in your risk of cardiovascular disease if you even got one of these. And obviously it increases dramatically, and especially those patients who develop type 2 diabetes. What we do know in PCOS, the majority of patients are obese. They're either overweight or, in fact, clinically obese with a BMI greater than 30. And what we also know is that the body shape of these patients changes. And you know, as a kind of a sports medicine institute, the difference between those patients who have got this kind of what we call this kind of the English side, which is very attractive subcutaneous fat around the hips and around the buttocks, or the kind of centropedal obesity that we see on the right-hand side. Both these female figures have exactly the same body mass index of over 30. But what you can see is that the waist-hip ratio on the left is 0.7, but here on the right is 1.2. And we know that an increased waist-hip ratio, one standard deviation of an increased waist-hip ratio will increase your cardiovascular risk and risk of cardiovascular death. And this centropedal obesity is basically made of seen of fat. And what you can see here is an MRI, and shown here in the white is actually all the actual central abdominal fat. And if one then just measures that, then what you can see is that intra-abdominal fat is directly related to waist circumference. We also know that 60% of these patients have insulin resistance, and insulin resistance is associated with increased cardiovascular risk. And what we also know is that those patients with PCOS at any given body mass index have a higher insulin resistance to normal patient or normal women. So what you can see is here that for insulin resistance is greater than those patients with who don't have PCOS, and you can see at any given BMI, then that insulin resistance is higher. And where is that insulin resistance? Where does it actually come from? Well, we think it's due to ectopic fat, and that is fat that resides actually in the liver, and therefore the fat in the liver then increases glucose output, or it's in the muscles themselves, and if you've got fat in the muscles, it decreases your glucose uptake. And that's really shown here in this kind of cartoon. And the thing that the increase in diacylglycerol actually is the main cause of this in the ectopic fat. And basically what it does, it increases the phosphokinase theta. Phosphokinase theta has a direct effect on insulin substrate 1, and as a consequence that decreases phosphoinositol kinase 3. In real terms, that decreases your GLUT4 receptors. Now, your GLUT4 receptors are critical for translocation into the membrane because these are the receptors that actually allow glucose to come into the cell. So you decrease the number of GLUT4 receptors, you decrease the amount of glucose, and by default, you increase the amount of glucose in the circulation. That's insulin resistance, and therefore you need more insulin to power that glucose into the cell. What we know is that women with PCOS have a 2 to 4-fold increase in either impaired glucose tolerance or type 2 diabetes, and it's most common if your body mass index is greater than 30. As I say, these patients are more insulin resistant for any one given body mass index. What we also know is that coronary heart disease, there's a 50% higher relative risk in coronary heart disease in these patients that are really not accounted for by traditional risk factors. And there's a lot of evidence out there in the cardiovascular risk markers that these patients have. But these patients are young. And as a consequence, if you're looking at genuine mortality, then you don't actually see it. There are a few studies which have gone forward looking at ladies in their 60s, and what we find is that there's an increased risk of ischemic heart disease, likely due to the accelerated atherosclerosis that occurs and as the ground has been set in their youth. So, what are the demographics of our Qatari population, these 13.5%? And what we can actually see is that they've got an increased risk of, they've got increased inflammatory markers, their cholesterol levels are overall high, their glycosylated hemoglobin as a marker of diabetes is actually increased. HDL is decreased quite markedly. Insulin levels are increased. Their testosterone levels are increased by the default that they've got polycystic ovary syndrome. Their pulse wave velocity as a marker of endothelial cell function is also increased, showing that these ladies have got increased arterial stiffness. And what's more, their systolic and diastolic blood pressures are increased. So these ladies are really a very metabolic phenotype in comparison to a Caucasian population where there are fewer cardiovascular risk markers for any given age group. What we also know, though, is that, for example, in pre-diabetes, the DPP study that was actually done showed that you can reverse it. So the most important thing that you can actually advise your patients is lifestyle. That is weight loss and exercise specifically. And there was a whopping 58% reduction in the cumulative incidence of diabetes with lifestyle in comparison to just treatment with metformin, where there's a 31% reduction in diabetes risk. What the PCOS Society, the Endocrine Society in America have said is that, well, there's not much kind of evidence there of exercise helping PCOS, but it's a good thing to do. Let's suggest that's a good thing to do. But what we do know is from those few studies that have been done with bariatric surgery, that there's a massive decrease in the hirsutism that these ladies have, a massive decrease in the testosterone levels, a massive decrease in insulin resistance. So we know that weight loss, substantial weight loss, is very effective. So what I want to do now, after that kind of brief introduction to polycystic ovary syndrome and why it's a very useful kind of population to actually explore as a marker for insulin resistance and the impact of exercise potentially, is to then take you through two small studies that have been undertaken. The first is in 12 PCOS patients with 10 controls. It was supervised moderate intensity, 60% maximal O2 consumption, 3 hours per week for 8 weeks. And we advise them to just continue their normal dietary habits. And this is a busy slide for which I apologize, but basically, those patients with polycystic ovary syndrome, their testosterone levels went down significantly, their triglyceride levels went down significantly, their insulin resistant levels went down significantly, and in both cases, with a glucose clamp, it basically showed their insulin sensitivity increased. And this actually was in the presence of their weight did not change. This was purely exercise, and therefore exercise alone had quite a marked impact on these patients with metabolic syndrome with PCOS. What I want to do now, though, is talk about a second study that we actually undertook, which was a lot more invasive, where we took 14 patients with polycystic ovary syndrome and 12 weight-matched controls. And in this, we actually took the patients and we gave them either, well, a 5-hour saline infusion, and the last 2 hours we did a glucose clamp. And then we brought them in again and did a 5-hour lipid infusion, and the last 2 hours were a glucose clamp, an insulin clamp. And the reason is because you're aware of the kind of the metabolic switch. We then exercised them exactly the same as before for, in this case, 3 hours weekly for 8 weeks, and then we repeated either saline or the lipid infusion over 5 hours to see what would happen. So basically shown here, kind of in this cartoon, where we actually took 12 subjects, normals, 12 PCOS, and either a saline or a lipid infusion, and we sampled them at baseline, at 3 hours and at 5 hours, and then supervised exercise. In fact, it was for 12 weeks in this case, and then we repeated again. And what in fact happened was, as anticipated, their insulin sensitivity improved, and that what we also saw is that their fatty acid utilization decreased quite markedly, and particularly their triglycerides went down with in the PCS group after exercise. So that in real terms, we didn't see that much despite the intensity that we actually put these patients through, which was very disappointing. However, when you infuse lipid, you increase insulin resistance, and that works by decreasing your glucose disposal. So what's shown here is the amount of glucose disposal that goes down if you infuse lipid. And shown here before exercise are the controls, and those patients with PCOS, there was 67% reduction in how you remove glucose from the circulation. But after exercise, both in the controls and particularly in the polycystic ovary syndrome patients, there was a marked improvement in the amount of glucose that was removed from the circulation purely by the fact that insulin sensitivity was markedly increased by exercise. What we're doing at this precise moment, we've also done the metabolomics on these patients. We're looking at all the fatty acids, and that's in the process of being looked at now, and you can see that there's a huge amount of data. And really, I just wanted to highlight this, because if there's the future of collaboration with the physicians and physiotherapists here at Aspetar, it would be excellent to actually look at metabolomics specifically and lipid utilization. So without further ado, I'd like to invite Manju to come up and talk about these new novel proteins that he's been exploring in the context of this study. Manju.
Good morning, everyone. My name is Manjunath, and I work as a scientist at Hamad Medical Corporation. For next half an hour or so, I'll be talking about the impact of exercise on recently discovered two myokines. Sedentary lifestyle have been previously demonstrated in various studies to show how impact on muscle mass. It's been shown to reduce muscle mass with increase in obesity. And obesity has been shown to promote macrophage infiltration in the visceral adipose tissue, which promotes chronic systemic inflammation. This further promotes insulin resistance and leads to various chronic diseases such as diabetes and cardiovascular diseases. However, exercise has been shown to have many beneficial effects, such as it induces physiological changes which benefits many organs. For example, as Steve mentioned in one of his slides, exercise has been shown to reduce the diabetic risks in individuals who have got high risk of developing diabetes by almost 50%. I think his slide showed 52, 58, probably this is another study. And also exercise also been shown to improve insulin sensitivity in these patients, reduce diabetic complications, control weight, and promote lower blood glucose levels and also cholesterol levels. And what are these potential mediators which would promote these physiological improvements? This could be some of the proteins which are secreted by muscle cells, which are called as myokines. And with the recent advancement in proteomics, we are able to measure many of these proteins which are secreted by muscle cells, so-called myokines. And these myokines can act as an autocrine, paracrine, or also act as endocrine factors linking skeletal muscle to physiological process in other tissues. And recent evidences shows that myokines modulate many metabolic parameters, reduces inflammation, and also improve biological processes. The first myokine which was discovered was interleukin 6, which is a famous, and till date it's the one which is quite widely studied myokine. And the study from Stensberg et al. Showed that they had a model where the human beings were advised to exercise on one leg and other leg was kept at a resting state. And during this exercise, they took the blood samples at various time points, and they found that the blood samples obtained from exercising legs had high levels of interleukin 6 compared to resting leg. And next, there's another study by Fabio et al. Which showed that when during exercise, there's an increase in plasma interleukins levels during exercise, and especially things like IL-6 went up, IL-10, and interleukin 1 also went up. This study also showed that how this interleukin 6 released from muscle influences various target organs which are important for obesity and diabetes, such as liver, then effects on adipose tissue and also on blood vessels. This study showed that IL-6, which are released from skeletal muscle, could inhibit. Is there a pointer somewhere? News. I mean, this study showed that IL-6, which is released from skeletal muscle, could act on adipose tissue, and it could induce lipolysis, which is a very big plus point. And this also showed that interleukin 6 could inhibit, it could promote glycogen storage in the liver, and it could also have block TNF-alpha induced insulin resistance in blood vessels. I mean, this was a review which nicely summarized the myokines, recently discovered myokines, and their effects on multiple organs. This one of them, quite interesting one, was irisin, which was discovered by Bruce Pigelman's lab about 4 or 5 years back. It was a hot topic at that moment. They called it as muscle exercise hormone. What they showed in the study was irisin, which is released from skeletal muscle, could impact white adipose tissue and convert them into brown-like phenotype. They also showed that it could this was able to do this by promoting a protein, key protein called UCP1, which is uncoupling protein 1, which is very important for brown fat activation and responses. And some of the other myokines discovered since then being IL-15, which has been shown to reduce fat mass by cross-talking with fat, and also it's been shown to promote muscle hypertrophy. And the other ones being BNDF, which is brain-derived neurotropic factors. And this protein promotes AMP kinase, as we all know that any drugs or agents which promote AMP kinase activates has got to play a role on fatty acid metabolism. And so they found that this protein increased fatty acid metabolism and also promotes muscle hypertrophy. And other one is FGF21, which is mainly secreted by liver cells, and this is a quite important hormone. It's quite hot topic at the moment as well. This has been shown to promote glucose uptake, and we published previously on this FGF21 from a previous lab where we showed that it protects the heart against ischemic injury, especially in obese hearts. This is another review which summarized some of all the fairly recently discovered myokines. The reason why I use this slide is I'm going to talk about one of the recently discovered myokine, myonectin, in next part of my talk. This is secreted by muscle cells with enhanced expression in skeletal muscle and also in circulation following exercise. This myonectin, also called as CTRP15, belongs to a family of proteins called CTRPs, which is C1q complement TNF alpha related proteins. And this protein family of proteins consists of about 30 secreted multimeric proteins which share a C-terminal domain homologous to globular domain of the immune complex C1q. And among this family members, the first one which was discovered was adiponectin. As we all know that adiponectin is a insulin sensitizer, and a lot of studies are being going on both as a clinical trial to use as adiponectin as a sort of molecule for diabetes. And CTRPs are the ones which have been fairly recently discovered proteins, and they are also important regulators of lipid and glucose metabolism. I would like to say this, there's hardly any studies on myonectin till date. There are very few studies. Every study is focused on rodent models, and there's no study in humans to date looking at metabolic aspects of myonectin. That's why we were interested to look at its effects in diabetes and other insulin resistant disorders. What this study showed was myonectin was mainly present, highly expressed in skeletal muscle at gene level, and both slow twitching muscle and fast twitching muscle has got myonectin mRNA being present in them. And they used C2C12 cells, which is a model for studying muscle physiology or pathophysiology. And they found that during differentiation of these muscle cells from myocyte to myotube, there's an increase in expression of this protein. So they used, a majority of these studies, they looked at as a model to study its function, and they used human embryonic kidney cells as a model to overexpress this protein in them, and they looked at the secretion, and they found that it was there in it was being secreted by the cells. This protein exists as multimeric forms in skeletal muscle and also in other tissues. On a Western blot or in a STS page, it migrates at a different intensity bands. One is highly glycosylated product, which comes around 50 kilodaltons or about 45 to 50 kilodaltons, and underglycosylated, which comes around 40 to 30 kilodaltons. And they also showed that this protein is being present in plasma in the mouse. So next comes there's another study from Peterson et al. Which looked at impact of exercise and obesity on this protein expression in mRNA and protein expression in skeletal muscle. They found first outcome from this study was they showed this protein is quite high in obese skeletal muscle obtained from obese rats compared to lean rats, Zucker rats. And when they induced both lean and obese rats on, put them on treadmill for about 9 weeks, they found there's an increase in gene expression of this myonectin in skeletal muscle. And later on, they looked at the protein expression in both lean and exercise model of the Secker rats, and this is quantified here. As you can see, that's with exercise, there's an improvement in myonectin expression. There's another study from Selden et al., which showed that exercise increases myonectin expression in both slow twitching and fast twitching muscle. And they put this was studies conducted in mice especially. They put them on a running wheel for 2 weeks, and they found that myonectin expression is increased in both in skeletal muscle and also in circulation. In the same study, they looked at the impact of obesity by using high-fat diet fed mice for 12 weeks, and they found that myonectin levels drop significantly with high-fat feeding in serum, and mRNA expression in the skeletal muscle is also significantly reduced in this high-fat fed diet mice. Next, they want to look at the effect of nutrition on this myonectin expression. Again, they went back to the same model, looking at slow twitching muscles and fast twitching muscle, and they looked at the effect of fasting and refeeding. What they did was they starved the mice for overnight and followed with 2 hours unconditional access to food. And what they found is upon refeeding, there is a significant increase in the expression of this myonectin in these mice, both in muscle and also in circulation. For some strange reason, the proteins of the CTRP families, most of them are quite high in females compared to males. But till date, no study has really looked into details. Why is that? I mean, you see, this is a common feature in most of these family members. And they used they infused glucose IP into these mice, and after 2 hours, they looked at the expression of myonectin in circulation, and they found that myonectin was increased in response to glucose, and they gavage fed same fasted mice with lipids, and they collected the samples after 6 hours, and they found that lipid also increases myonectin. And to confirm these findings, they went back to the skeletal muscle model. They used this L6 cells, and they cultured the cells in presence of glucose and palmitate, which we usually use as a fatty acid model. And they found that these results were able to mimic in a cellular model as well. So for the previous studies have shown in rodents have shown that myonectin is highly expressed in obesity. Exercise promotes myonectin expression in circulation and also in muscle. Myonectin is regulated by nutrition states. Therefore, our hypothesis was to understand the importance of this protein in regards to obesity and insulin resistant states such as Pcos. So we did this by measuring the circulating levels of this myonectin in control versus Pcos subjects, and also we studied the effect of metabolic challenge and how it affects myonectin expression. This was the same study which Steve presented, the same samples from his studies. And our experiment result was we had 12 control subjects and 14 Pcos subjects. They were given either saline for 3 hours, saline clamp followed with the 2 hours of insulin infusion, and or with the lipid infusion followed with insulin clamp for last 2 hours. And they were allowed to exercise for about 3 weeks, supervised exercise for 3 months, 3 days a week for 1 hour each day. And following that, the same protocol was repeated. But the data what I'll be showing is only with lipid clamp and insulin, because we didn't find any significant changes with saline and insulin clamp. So what we found in this study was when there was insulin resistance induced by lipids, there was a significant drop in myonectin levels. This mimics what they found in mouse model in Selden's paper. And this was effects were negated by introduction of insulin clamp. Basically what we are saying is when there's a metabolic switch, there's a alteration in myonectin expression. We found similar response both in control patients and also in Pcos patients prior to exercise. And these are the values which shows mean and standard deviation of the values we found from the study. And similarly, following exercise, we did the same measurements in the samples obtained after exercise, and we found that now once following exercise, when there was improvement in insulin sensitivity in these control subjects, we didn't see same level of decrease in monectin levels compared to what we found before exercise. However, in PCOS subjects, we saw some drop, but which was not significant. But the recovery was slightly more than what we found in baseline levels. So next part, we wanted to look at what does how the levels of this myonectin in PCOS or control. I must say this is very preliminary data what we have, because the sample size is very small, and within this, with small size of samples, about 10 and 11 samples, we found that there were significant levels of myonectin in PCOS patients. Also bear in mind, these PCOS patients are generally obese, and majority of them, almost 60%, are obese. So this could be reflecting either condition of PCOS or could be obesity itself triggering the increase in expression of this myonectin. However, we didn't find any great change in terms of myonectin response either before and after exercise, both in control and also in PCOS subjects. This could be because of our sample size. You can see a trend of increase, but again, the sample size is quite small. If there's opportunity to do large cohort, probably we'll find a better outcome, I guess. Just to conclude from this myonectin study, what we found was myonectin was significantly higher in PCOS subjects compared to control, and lipid challenge decreased myonectin expression both in controls and PCOS subjects. And lipid-mediated reduction was negated by insulin infusion, and lipid and insulin inversely regulate myonectin expression in plasma. Exercise induced a small but insignificant change in myonectin levels in PCOS subjects. The ongoing work, what we are trying to explore is now to evaluate the circulating level of myonectin in metabolic syndrome phenotypes such as obesity, diabetes, PCOS, and NAFLD in large cohort studies in collaboration with Professor Steve and others in Qatar. Also, we would also like to, if possible, we could engage Aspetar. We would also look at effects of exercise, high intensity versus moderate exercise on circulatory and salivary myonectin levels. We have some preliminary evidence in very small population that saliva also produces myonectin. And also we would, if possible, we would also like to look at effects of endurance versus resistant exercise in on circulating myonectin levels in obese individuals. For the next part of my talk, I'll be talking about another recently discovered myokine. This, till date, there's only 2 papers on this protein. This protein is called as MOTC. It's a mitochondrial derived protein which was discovered in 2015 by a group from University of South California. I mean, both the studies are in rodents, and we are also the first study which is looking into human aspect of this protein. This MOTC stands for mitochondrial open reading frame of 12S ribosomal RNA-C. It's a very small peptide, 16 amino acid in size, and it's released from 12S ribosomal region of ribosomal RNA region of mitochondrial DNA. And what this seminal paper showed was MOTC is expressed in brain, heart, muscle, and testis, the key metabolic organs, and the levels drop during fasting in skeletal muscle, and the MOTC is present in plasma, and the levels are dropped during fasting. It's a complex data, but I'll try to simplify. What I would want you to focus is just on this bit. What they've used, sorry. Oh, it's not sorry. They've used HEK293 cells, which is human embryonic kidney cells, as a model to study its function of this protein. They overexpress in them in these cells, either stably expressed or use this as a drug to stimulate the cells. And they looked at transcriptome analysis. What they found is the genes related to acetylcarnitine or methionine or TSA cycle were upregulated when the cells were overexpressing this MOTC. However, the genes which are related to purine synthesis and dipeptide synthesis related genes were downregulated. That's the only take-home message from this busy slide. Okay. Based on their previous studies in 293 cells, they employed this mass spectroscopy approach to look at key metabolites produced in stably cells overexpressing this MOTC. And what they found is MOTC promotes glycolysis, inhibits this methionine foliate cycle, leading to accumulation of a key product within as a metabolite in the cells, ACAR. ACAR is a drug which has been known to stimulate AMP kinase, and any drugs which activates AMP kinase has got to have a role on fatty acid metabolism. That's what they were trying to address in this study. And as they expected, what they found is the ACAR levels were almost 20-fold increase in the cells overexpressing MOTC. And the cells overexpressing MOTC or cells which are stimulated with MOTC, they found that there's increase in AMP kinase activity. And they found increase in acetyl carboxylase CoA and CPT1, the two key enzymes which are involved in fatty acid metabolism. CPT1 is a transporter of fatty acids, and ACC is a very important protein for fatty acid metabolism as well. And you could recall from Steve's slide that GLUT4, which is important for glucose utilization, was also upregulated in the cells overexpressing MOTC, in the cells stimulated with MOTC. And this, again, they showed time response, how they change with respect to doses at 8 hours and 24 hours. Right. Then they went to look at whole animal model. They used mouse as a model for this study, and they used obese mice. They fed the mouse with high-fat diet for 12 weeks, and they looked at insulin sensitivity, whole body insulin sensitivity, and also glucose response. They were looking at this study. What they found is they gave about 5 milligrams per kg body weight MOTC to this mice for 1 week, once daily IP. And what they found is on glucose tolerance test, the mice which received MOTC treatment were able to clear glucose faster compared to wild type control mice. And glucose infusion rate was also much, MOTC stimulated treated mice required more glucose, and the glucose infusion rate was much higher, which reflecting whole body insulin sensitivity was much higher in these mice compared to wild type mice. Next, they looked at insulin stimulated glucose disposal rate, which primarily reflects skeletal muscle insulin sensitivity. And they found that mouse which received MOTC treatment required had better glucose disposal rates. However, they didn't find any changes in hepatic glucose production suppression in both the mice. And they also found in MOTC stimulated mouse skeletal muscle there was high amount of AKT phosphorylation, and MOTC levels were much higher compared to wild type control. Next, they want to study as we know that with aging there is an impairment in insulin sensitivity, and they used a mouse again as a model to study this. They looked at 4-month-old mice and 32-month-old mice. They took the skeletal muscle and looked at the MOTC expression. The MOTC expression was much lower in aged mice compared to younger mice. This is we quantified here. And what they did was they used this. They first initially compared the insulin response, glucose uptake in this both the mice, and they found that glucose uptake was much lower in aged mice as expected. And they injected this mice for 1 week IP injections of MOTC, and they found that when the MOTC was injected into this mice for 1 week, there was a reversal of effects. You could say there was an improvement in insulin sensitivity, and glucose uptake was much better. To confirm this, they used cellular model L6 cells. They differentiated them into muscle cells, and they incubated the cells with media containing high glucose. At the end of 24, these L6 cells, one batch was just control cells, another batch was cells overexpressing MOTC. And what they found is the cells which overexpress MOTC had utilized most of the glucose in the media compared to control cells. So from this study, what the hypothesis was, they hypothesized that MOTS-c is released from mitochondria in response to aging or stress, and MOTS-c inhibits de novo purine synthesis, activates ACAR and AMP kinase pathway. Because of this AMP kinase, it's able to promote mitochondrial metabolism, fatty acid oxidation, and glycolysis. Thereby, it restores metabolic homeostasis. And the study also showed that skeletal muscle is the main target for MOTS-c, and MOTS-c promotes metabolic homeostasis in both high-fat diet mice and aged mice. So, as I mentioned, there's only 2 studies on MOTS-c, and both of them are on rodents. And what it showed is, as I summarized before, MOTS-c protects mice against insulin resistance induced by fat diet or aging. So our hypothesis was to look at this MOTS-c in pathophysiological states associated with insulin resistance, such as diabetes, obesity, and Pcos. So first we looked at, first of all, till date there's no data about MOTS-c expression in humans. So we used a panel, cDNA panel, to look at its expression in various tissues. Then we looked at whether this could be a potential biomarker for diabetes or Pcos. And third part of the study was to look at MOTS-c level in response to metabolic challenge. As expected, because of mitochondrial origin, the MOTS-c was fairly expressed in most of the cells, most of the human tissues we looked at. And metabolic tissues such as heart and liver had the highest expression of MOTS-c at gene levels, compared to skeletal muscle, pancreas, or placenta. And next, we looked at circulating levels of MOTS-c in pre-diabetes and diabetic subjects, and we found that very significant. Even again, our sample size is quite small here. It's about 20 and 20 in each, I think. So within this small population, we were able to see a significant increase in MOTS-c level in diabetic subjects compared to pre-diabetic subjects. Next, we want to look at how does MOTS-c correlate with insulin sensitivity, and we found that MOTS-c significantly but negatively correlates with insulin sensitivity, and also negatively correlates with steady state plasma insulin levels in these pre-diabetic subjects. So this for our Pcos study, we used the same model which I showed before. And what we found with MOTS-c was, unlike myonectin, which reduced with lipid challenge, MOTS-c expression was increased with lipid challenge. In other words, insulin resistance promotes MOTS-c. This has also been seen in diabetic patients in our diabetic samples. This effects of lipid infusion was negated by insulin. It was almost brought to near the basal levels, both in control and also in Pcos subjects. However, if you look at this level of significance, it was much highly significant in Pcos patients compared to control subjects before exercise. What this reflects is Pcos subjects were already insulin resistant. When you further challenge them with lipids to induce further more insulin resistance in them, there's a high amount of MOTS-c expression. And following exercise, we'll again compare MOTS-c levels in control subjects and in Pcos subjects. But now, in control subjects where there was an improvement in insulin sensitivity following exercise, there's no though you can see a bit increase, but it's not statistically significant increase what we are seeing here in control subjects. Whereas in Pcos, we still see that response, there's increase with lipid challenge, which was more or less negated by insulin. Next, we want to look at if there's a difference in MOTS-c expression between controls and Pcos subjects. We didn't see, though you can see slight increase in mean MOTS-c levels in Pcos, but they were not statistically significant. This could be because of reflecting this one patient which had very, very high levels of MOTS-c. And exercise, again, we didn't really see any change impact of exercise on MOTS expression in this small study. Just to conclude on our diabetes and PCOS studies on MOTS, we found MOTS is significantly higher in diabetic subjects compared to pre-diabetic subjects, and insulin sensitivity negatively correlates with circulating MOTS in pre-diabetic subjects. And from our PCOS study, we found that lipid challenge increases MOTS levels both in control and PCOS subjects, and insulin resistance induced through lipid infusion promotes circulating MOTS levels, and the increase was much higher in PCOS compared to control. And lipid, we also demonstrated from this small study that lipid and insulin inversely regulate plasma MOTS levels in vivo. And the studies in progress, we are looking at effects of, circulating levels of MOTS in metabolic syndrome phenotypes. And as a part of other study in collaboration with Professor Abu Samra, we're looking at impact of insulin resistance on skeletal muscle MOTS expression in human subjects. And the other studies we're also looking is compare the effects of high intensity versus moderate exercise on circulatory and salivary MOTS levels. And finally, if there's a possibility for collaboration, we would like to evaluate the effects of endurance versus resistant exercise on circulating MOTS in obese individuals. This leaves me to thank my people who really helped me on these studies, especially Professor Steve Atkin provided me with lots of samples and guidance on the study. Abu Samra, with whom I work with, Professor Ramzi, who's director of our institute, and our research team, and Professor Satya, who sent us samples from UK, and the source for funding, MRC, and ITRI, where we work in. And thank you very much for your time.